Thank you again for the invitation to speak today. It's a real pleasure to be here. So I'll be sharing some results from, again, the NeuroCovid-19 studies focusing on physiological and functional brain changes in adults recovering from COVID-19. So over the past year and a half spent in the pandemic, which is pretty hard to believe now, we know that COVID-19 manifests primarily as a respiratory illness. We also know that in certain individuals, typically those with more severe courses of illness, that multi-organ injury is possible. And so on MRI, for instance, abnormalities are present not only in the lungs, but also in the brain, the heart, liver, and kidneys as well. And one organ that warrants special attention is the brain, and Brad did a great job of providing an on-ramp for this. And although the exact mechanisms by which SARS-CoV-2 affects the brain are not fully understood, we do know that morbidity due to neurological and psychiatric symptoms is quite high, even in those recovering from infection. One particular study that I'd like to highlight, reference 3 here, found that among nearly 250,000 individuals with COVID-19, the estimated incidence rate of a neurological or psychiatric diagnosis in the following 6 months was about 33%. And so more work is needed to uncover brain-related changes associated with COVID-19, especially in those that are recovering from acute stages of illness. And so neuroimaging has played an important role in describing neurological events in those with COVID-19, pointing out features like encephalopathy, punctate lesions, such as microbleeds or white matter hyperintensities, as well as stroke. However, less is known regarding physiological and functional brain changes that are associated with COVID-19. And as can be seen on this abbreviated timeline of COVID-19 neuroimaging publications of this past year, only a few studies denoted in bold font report on brain physiology or function. And while this slide does not include all neuroimaging studies of COVID-19, I think that would be pretty hard to fit into one slide. I think it does reflect the proportion of studies that focus on structure alone versus interrogating additional aspects of brain health. Oh, whoops. And so, yeah, in this study, we sought to investigate physiological and functional aspects of brain health in adults recovering from COVID by leveraging the comprehensive brain MRI protocol collected from the NeuroCovid-19 study. And today I'll be discussing 3 pulse sequences from the initial assessment, whose details are shown on the right side of the slide. And so in the top row, a 3D P-Castle sequence was used to yield measures of cerebral blood flow. In the second, or in the middle row, a bold sequence was used to extract the fractional amplitude of low frequency fluctuations, or fALFF, and this is a proxy measure of brain activity. And then finally, in the bottom row, a T1 weighted sequence yielded measures of gray matter volume. And so in the analysis that I'll be presenting on today, there were 56 adults included and sorted into the 3 groups that are shown along the bottom. And Brad introduced these groups, but I'll just run through them really quickly. So the first group comprised 13 control participants that were negative for COVID, but they did present with flu-like symptoms. The second group had 35 participants positive for COVID and who self-isolated at home. And then finally, the third group comprised just 8 participants that were positive for COVID and who were hospitalized as a result. Now, to wrap this slide up, I'll just highlight 2 important aspects of the study that I'd like to make a takeaway, I guess. And the first point is that we focus on recovering adults that are about 3 months beyond infection, so well past acute stages of illness. And then the second point is that we use a multi-contrast imaging protocol, of which a few of the sequences are shown here. And so using this data from the initial assessment of the NeuroCovid-19 study, our aim was to compare CBF and fALFF in adults recovering from COVID-19 relative to controls with flu-like symptoms. And our hypothesis was that there would be differences in CBF and fALFF between COVID-19 and control groups. And I'll note that in this first pass analysis, we chose to combine both hospitalized and self-isolating participants into just a single COVID-19 group. And this was mainly driven by the smaller sample size we have of hospitalized participants. So to test these hypotheses, we performed voxel-wise comparisons of CBF and fALFF by following 3 steps. So firstly, data was registered to a standard space. Then a general linear model was created to test for group differences while accounting for age and sex covariates. And then finally, cluster extent thresholds were estimated using tools from AFNI. And so in the next couple of slides, I'll be presenting the results from this analytic framework. So starting with the CBF data, here is a series of axial images that depict a single cluster of significantly decreased CBF in the COVID group relative to the control group. And this cluster encompassed several different regions, including the orbitofrontal cortex, the thalamus, and regions of the basal ganglia, including the caudate, the putamen, and the pallidum. And individually, these regions are important for several different functions, but altogether, they actually represent key regions of the cortico-basal ganglia thalamocortical loop, which I've shown just a small diagram in the bottom right here. And this is a network of brain regions that are involved in complex behaviors, such as reward-based decision-making, affect regulation, and goal evaluation. And what's interesting is that this network is also quite relevant to several neurological and neuropsychiatric conditions, which could relate to reported COVID-19 symptoms such as brain fog or fatigue or anxiety. To further compare these findings with the literature, the closest match to our data and methodology is found in PET studies of glucose metabolism. And so on this slide, I'm just showing results from 2 PET studies that both show hypometabolism in adults recovering from COVID-19 compared to controls. And by and large, many of the regions that we observed in our own analysis seem to map onto those that are seen here, including the caudate, the thalamus, and to an extent, the orbitofrontal cortex. Decreased cerebral blood flow and hypometabolism through neurovascular coupling could be evidence of reduced neuronal activity. And so altogether, we can speculate that these regional findings map onto certain aspects of COVID-19 symptomatology. So moving to the fALFF data, which again is a proxy for brain activity, we observed 4 different clusters of decreased fALFF in the COVID-19 group, encompassing several frontal and limbic regions that I've labeled here on the slide. And in comparison to the CBF analysis, these clusters are slightly more widespread, but they do partially overlap with some of the same regions and may thus point to similar COVID-19 effects, possibly related to reduced brain activity along established networks of the brain. And to try and frame these results with previous literature, and I'll just reiterate here that there are not many functional neuroimaging studies of COVID-19, I do want to highlight one case report of 2 COVID-19 participants that presented with anosmia. So here I'm showing patterns of functional connectivity in one of the COVID-19 participants on the top row, and then one control participant in the bottom row. And we can see that the COVID-19 participant exhibited widespread decreases in functional connectivity, especially in proximity to sort of inferior frontal regions, such as the orbitofrontal cortex. Now, to support this small case report, others have actually found similar functional connectivity deficits in those with non-COVID-19 related anosmia within established networks responsible for olfactory processing. And so in future work, it'll be interesting to connect the imaging-derived phenotypes of CBF and fALFF with cognitive and behavioral measurements, which have been collected in the NeuroCOVID-19 study, as Brad alluded to in his talk. So I finally talked about gray matter volume as another imaging-derived measurement in the first couple of slides in this presentation. And for completeness, we ran this data through the same analytic framework as the CBF and fALFF data. But in this case, we actually did not observe any between group differences. As it turns out, there are mixed findings on this imaging phenotype in the literature. So for example, one group found that there were no differences in gray matter volume in COVID-19 participants about 2 to 3 months post-infection when compared against control. So quite similar to our study. Another study, however, observed greater gray matter volume in COVID-19 participants, again, about 3 months post-infection relative to controls. And then one final study I'll highlight showed longitudinal decreases in gray matter volume and cortical thinning in a larger sample of participants for whom pre and post-infection data was available through the UK Biobank. And so these somewhat diverging results could be due to a number of different factors, for example, timing of assessments or cross-sectional versus longitudinal data. But needless to say, more work is needed to paint a clearer picture of this imaging phenotype. So that was my last slide, and I'll just summarize here. We observed CBF and fALFF differences between adults recovering from COVID and controls with flu-like symptoms. These changes were observed in regions that have been highlighted in previous neuroimaging studies of COVID-19, and they may also underlie COVID-19 related symptoms such as fatigue, brain fog, or anxiety. And I'll note again that this was a first pass analysis, but moving forward, there are many different things we'd like to try. But I think the most important next step is to look at longitudinal change in this sample of adults who had a follow-up assessment about 3 months after this initial assessment. And so hopefully we'll have some results to share soon. So I will end by just thanking all research participants in the study and collaborators listed here. Thanks for your attention.